Next gen, we talked about Human Genome Project, platforms, like which instruments are out there. We talked about the assays yesterday. I mean, sorry it's such a sort of snowstorm of things, but that's sort of the way next gen is. So today is the last missing key piece of the next gen process, which is the analysis. And I don't promise that you'll come out of here being experts in this, because I'm not either. I'm still trying to play the game and learning this. And to be honest, I've only been doing this for a year and a half. And I'm not a bioinformatician by training, but I kind of get the sense of what you need to have in place to do the analysis and how to think about it in terms of a clinical assay. So with that, let's say we do our 500 gene capture panel and we send it off for sequencing. Alternatively, you can have a sequencer in-house as well, so that's another debate, insourcing or outsourcing of sequencing. We send it out, and within 10 days, 2 weeks, we get the data back. And typically for a 500 gene panel, you might get back, depending on the coverage that you get, anywhere from 5 to maybe 10 gigabytes of information. So 500 genes. We're going to go into some of the informatics here. 500 genes, you're covering, say, roughly 20 exons per gene. So we're talking about 10,000 exons, right? And we want the coverage to be roughly, let's just make it easy for now, a mean coverage of 200x full coverage. Okay. So you have this much amount. I think it equates to probably 4 megabases of information. So 4 million. So if you do the math here, you'll have 8 times 10 to the 8th, right? Of data. And what is that? So about 800 megabases or so. Will give you about this average full coverage. I'm just throwing some numbers out. So this part will be maybe on the lower end, about roughly 5 gigabytes of data. Okay. And if you do a tumor versus normal, you would want to double all of this, right? Times 2 times 2. And this would be 1.6 gigabases of information, roughly 10 gigabytes of information. So this is what you're going to get back. And with this amount of data, just to quickly go into the informatics a bit, if you did, say, 50 samples or something, 50 samples, you're looking at about 500 gigabytes of data. That's a lot of data. If you're sitting at home on your Verizon Files, right, or Comcast, Xfinity, and you try to download this amount, I think it would take you a good week or two. So the best option to receive this data is to package it into a hard drive, an external hard drive, and they just ship it to you by FedEx. That's what most people do nowadays. So this is just to tell you that the data is quite heavy and intensive. And this is just the raw data, by the way. This is just the FASTQ files that you get out, and I'll explain what that is in a second. So when I talk about next-gen sequencing, the thing I worry and I'm most scared about is the informatics. And this is one component right here in terms of the analysis, is the storage. So one problem is storage. The second problem is doing the analysis itself, right? The analysis in terms of doing this process takes a lot of computing power. If you were to try to take this 10 gigabyte file and run it on your desktop right now, your partner's workstation might have 2 cores of CPU computing power. Actually, it won't even run because you only have about 2 gigabytes of RAM on it. For the fastest mapper out there, you need about 3 gigabytes of free RAM to accommodate the human reference genome. And then thereafter, a little bit more to make sure your computer is running. So let's just say you have your 2 cores of processor and you have 8 gigabytes of RAM, enough to do this mapper. On that computer, it'll probably take you probably a few days to run, just to map, a few days. If you give it a little bit more computing power, say 8 cores or 12 cores with that amount of RAM, it'll probably complete within 2 hours. So it takes a lot of computing power to do that. And that's one case, by the way. So if you had 50 of these cases, you would need about 50 of those computers running, right? All in parallel at the same time. So the solution is not to have 50 computers running. The solution is to make your computers denser with processing power. And that's why you need a cluster. So computing is another problem in terms of NGS analysis. So clusters, what they are, just to briefly go into that, is we're used to having like a PC, right? There's your PC, or personal computer. What you could do is actually wrap this all into one blade of a cluster. And in a blade, you can have maybe 2 processors going on at the same time. Each one can have, say, 8 cores. And each one of these can have 16 threads. You don't really need to know all of this, but I'm just illustrating to you what this is. And it could have, say, 96 gigabytes of RAM. One of your computers would have 2 gigabytes of RAM, maybe 2 cores nowadays, like a dual core processor. So you can see that you can, this one thing here is the equivalent of about 8 or 10 of these smaller computers. And you can wrap it into one. And it's smaller than your desktop. It's basically the size, I don't know, yay, wide, that high, and that deep. So it's like your VCR player, maybe 2 or 3 times that size. This is how big. Oh, can't believe I say VCR player. DVD player. So you have the size of 6 of your DVD players, right? Because it's that big. Yeah. But VCR is about that big. Yeah. I swear I don't play VCR tapes anymore. So it's about that size, and the reason why to do this is because you don't have, 10 of these PCs would probably take half of this room, right, to put in. It's a space issue. You have 10 of these with 10 different power supplies, which generates 10 times the amount of heat. So this one, you only need 1 power supply, is 1 rack. But what people do to make this even better is they put in 10 of these. So you can imagine now, instead of having just 10 of these computers, you have 100 of them. And this will fit in the size of this cabinet right here. And that's what is called a cluster. So they all stitch together so that they share sort of the same processing power. You can distribute jobs across the different nodes. So this will be 10 nodes. Each one will be a node here. And this will cost 100,000, 150,000 to get that amount of computing capacity. So this is just computing, by the way. So that takes care of that portion.
So you have that in the budget?
Yes, you would definitely need that in the budget to make this work.
So do you put one of those clusters separated by 1 computer, or is it,
What's
how
that?
does it work? The cluster, do you just like kind of plug it in 1 computer and you're done, or does it need to be?
So the way this will work is you have a huge power supply down here, way more than what you would have in a wall outlet. So it needs like access to, I think, 230 or 220 volt either way. So
Sure.
you need to get B&G to come in and install enough power to run this, because this is a lot of, each one would probably be sucking up 500 to 900 watts of power. So it's like running 10 blow dryers. You can imagine if you ran a blow dryer and just a toaster at home, right? That's 2 of these, but you have to run 10 of them, and it will, trip your circuit on a regular circuitry. That's why you need hefty power at the bottom, and then you just have stacks and stacks of this, like this. And if it's eating this much power, well, what is the problem that you see here? It's heat. So energy in has to escape somehow out, right? And so some of that gets, moves the electrons or whatever to do the computing, but most of it ends up being heat. So you need a lot of ventilation and cooling power, and a door that is open, maybe, or even some people put together some cooling sort of system, refrigerant system next to it to keep it cool. But all this is housed together into one unit, compact, so that it can all be next to each other, so you need to transfer data back and forth. It's very, very efficient. And what else is there? So now that's just the computing, right? So the storage will have to be somehow attached to this. So people typically do a network attached storage server. So this could be another box sitting in the same room, or it could be in a separate location that you need. Let me erase some of this. It's a bit more into computing than I had planned to do, but maybe this interests you. You have a network attached storage server running some form of, let's say, a RAID 6. A RAID 6 is redundant storage that allows you to have some failure in terms of your hard disk. So if you just store your data on a disk, right, who knows? That motor in there runs out, and then you're dead. I mean, you can't recover your data anymore. But what a RAID system is, it's an array of disks. So you can have 10 disks on here, somehow hooked up to each other, and they share data across the disks. So you spread data across so that if you have one failure in a RAID 5, you'll be okay. You have redundant data across the other 9 disks to help you rebuild the array. But even better is a RAID 6. You can have 2 failures, and you can still rebuild the array. The problem with a RAID 5 is during the rebuild, if a second one fails, you're screwed. You've lost all of your data. And each one of these can be 2 terabytes each. So if you have 10 of them, you have a 20 terabyte RAID, but you need to subtract, for RAID 6, 2 of those disks. So now you're down to 16 terabytes. So you sacrifice some storage so that you can get redundancy, and this is very, very important. And so you have this storage system here. Operationally, for the amount of cases that we're doing, for this amount of data size, for the 50 cases a week or so, I think we need basically a 3 to 6 month window of data. We would probably need on the order of 200 terabytes of data storage. And this will be the online storage. So immediately accessible. So you can say, go in, pull this case out, put it into the computing system, the cluster, do the analysis, save the files, push it back to here. This is only for computing. It won't be storing any data if you use a network attached storage server like this. The other problem is hurricane comes into Boston for some reason. It hits Jackson 10 and knocks out our cluster. That's bad, right? You need another mirror of this. Okay? Like, in another building, maybe in Warren, in Charlestown. Charlestown is closer to the water, so that might be worse. But still, you need 2 physical locations so that unless something happens to the entire city of Boston, you will at least maintain one of these copies right here. So this will be mirroring this. It will be a mirror. So this will be maybe near line storage. So if this fails, we can rebuild this back very quickly by just transferring data. That's a lot of data to transfer. But then with clear regulations and pathology and clinical testing, there might be a rule that you have to keep this data around for 5-10 years. You know, slides you're supposed to keep around for 10 years, right? If there's a rule, such rule, and we can't trash any of the raw data, that means you need a offline storage. So possibilities are you convert this into tape, you put it into a box and you send it to a place like Iron Mountain or Retrievax, and they'll store this in their facility. It'll be crazy to have to recover this data. But you need some cheap solution here, because over 10, maybe 5 years or so, or maybe even 3 years of operation, we're looking at about 1 petabyte of data. That's a lot of information. So this is all the sort of nuances of the computing system. There are other ways to represent this. You can actually put it as part of the cluster as well, which is called distributed computing. It's sort of the model of Facebook, Google, Yahoo. They use this. It's a very different way of storing data. But the other thing that is not apparent into here is that because you're working with so much data, the pipes that are transferring data have to be very fast, right? I mentioned about Verizon and all that. Here you probably want a minimum of a 10 gigabit connection. So 10 gigabit or 10 gig E connection. This is way faster than what we have here right now. Your thing right now is probably 1 gigabit, but you probably don't even get that much speed or capacity. But a 10 gigabit will be able to transfer this file probably the 10 gigabytes or so in 1 second. Actually, no, I'll take that back. 1 gigabit divided by 8, probably 10 seconds. Take about 10 seconds to transfer that. So you need these fast connections between Charlestown and here, which there is a pipe, but is not being fully used right now. You need, let's say your sequencer is in another building and you need to transfer, you have to connect all the pieces together with that fast enough connection. So networking is another complexity up here. And then finally, once you analyze the data, map it or whatever, the interpretation is the other complexity, right? You're going to get a lot of unknown calls, and how do you make sense of that, et cetera, et cetera. So these are the different challenges I see with the analysis sort of pipeline. So this is the infrastructure, right? So I talked about, I think very briefly yesterday or the other day about infrastructure. The wet lab needs to be in place so you can do library construction, you can do capture, you should be able to automate it so that you don't have 10 techs. BB can attest to this. Making 30 libraries is not fun by hand, so you need automation for that. But then on the computing side, you also need the infrastructure because this requires probably 1 full-time IT person to help maintain. And then the analysis itself and the interpretation takes 2 or 3 bioinformaticians and people to make sense of the data. So we are not there yet right now with the infrastructure. But yeah, that needs to be in place. Yep.
So since computing power is so important, is anyone actually thinking of designing a chip that just physically in the hardware is optimized to decode AGCTs? It's like map it. Instead of using like software instructions, it itself intrinsically understands that.
Right. So you're talking about the you do your sequencing, right, and then it spits out an image of those dots. You want to bypass this somehow?
Either that or like with the mapping part too. I mean.
Even the mapping part.
Yeah, you're obviously converting that to like zeros and ones or something computer understands. I wonder if there's, and I don't know about computing, but like if there's some way it's hard to encode that.
It's possible because that's always the fastest, right? There's no intermediate steps of transfer, and so what Alice is talking about is you do your sequencing. And bam, you get a mapped file out directly from the sequencer. In the future, that might be the case, where you don't have to transfer the data somewhere else to have it interpreted. It's almost like, what's the best example I can give? A glucometer, something that measures glucose. Today, you insert a strip, bam, in the display, you see the glucose level, right? So the analogy of what we're doing right now is you put the strip into there, and then the glucometer spits out this other thing that you have to insert into another machine to interpret it. We're kind of not there yet with next-gen sequencing because it's not as automatic, but one day, hopefully, that'll be the case, is that it's all built into one box, right? So that you get out the data. So that's almost like a point-of-care kind of testing with next-gen sequencing. People are talking about this. Already, like with nanopore sequencing. But I think the massive amount of data so far, nobody has quite a clear-cut solution for that quite yet. So that's all the infrastructure that is needed for computing. And you guys understand why it's so crazy and such a beast to deal with? So we get back that 10 gigabytes of data. Originally, it would have started as, let's just say it's an Illumina run, right? It will be, during the sequencing, the Illumina would generate the images. So if you call back to the lecture on the platforms, it's sequencing by synthesis. You add on reversible dye terminators that you image across. You shine a laser on it, it shines some color saying that it incorporated an A, a T, C, or G. So you do this for various cycles. You can do, say, 100 paired end run. What this means is that you sequence your fragment in this direction, and then you take that away, and then you sequence in the other direction. So you get a paired end. The two ends are paired together It's actually 2 reads, but they are paired reads, and we do 100 bases on each side. That's what 100 paired in means. Some other folks do 50 base paired in. Nowadays with MySQL you can do 150 bases paired in. There are different reasons to doing paired in, but let's just say you do a paired in run. So this would have undergone 200 cycles of sequencing. 100 in one direction, 100 in the other direction. So it's like doing a forward and reverse sequencing. So what you do is you capture all of these images, and I don't think there's enough computing power that I don't know if it really analyzes in real time. I forget the strategy, but at any rate, if you were to store all of these images, it takes this over 10 days. I think from 2 flow cells. So remember, HiSeq 2000 has 2 flow cells going. Do you know how much?
45 terabytes.
Yeah, it's somewhere between like 35 and like 40 terabytes, something like that. It's huge. So much that this will completely max out my 200 terabyte thing, right? So that you basically just trash this. You take this data, and going back to the glucometer kind of analogy, you have to go through another step that will take the image file and push it into FastQC format, or FastQ format. And this goes from 35 to 40 to about 1 terabyte. And this is just basically a text file. Your AATs, C's, and G's, etc. So it's still a lot of data, right? It's a huge amount of data. This is equivalent to 200 to 400 gigabases of information. And that's how you're going to fit those 50 patients and more onto this run. So you get this back, and so what is FastQ? In the old days, a FASTA file is essentially a name of a sequence, and then your A, T, C, G, C, C, A. Something like that. A FastQ looks a little bit fancier. I think there are different characters that are built into here, but essentially it has a sequence name again. It has sort of the basis here, but included in here is a quality score. And I forget how it's encoded, but there are some characters in here, the weird characters. It might be a question mark, an exclamation point, an @ sign. But these are going to say what the quality of each of this base is. And that's what is added on top of the FASTA format. Why do you need this? So you need this because during the sequencing step, the sequencer knows what quality of signal was taken from that image, how bright it is. So let's say here's a circle, right? It's going to say, oh, how bright the signal is, how large the signal is, maybe over time, like how brightly it shines or whatever. So a combination of this, they have modeled within their system to say, reliably, if it's this bright, I can, 95% of the time, I'm correct if I call this an A. If it's this weak or that poor, 50% of the time I'm correct. So they use that as a way to build in a quality score to say, what are the chances that you are correct when you call this A? What are the chances that you are correct when you call this a T? And that's figured into a Q score, or a quality score. And the quality score is based on a Phred score. And this is back from the human genome sequencing days, where they had to come up with some system for Sanger to say how good are my my blips and my peaks when I'm doing Sanger sequencing. So what this is if you have a Q20 score, what it means is that you have a 1, should be an arrow here, over 10 to the Q score divided by 10 chance of having an error. So if you take 20 here, this will be 1 over 10 to the second, right? Which means it's a 1%. So that means that if I have this A and it's a Q, let's say this character here is the sign here, and that means it's a Q20. This base that I'm looking at, 1 out of 100 times, it's not going to be an A. It's going to be something else. If I have a Q30 score, it is 1 over 10 to the third, right? 1 over 1,000. This means that 99% of the time, 0.9% of the time, I'm going to be correct when I call this T right here. That's pretty good, right? So Illumina sequencing tends to grade around Q Q30 to Q40. But in the industry, people tend to think they overestimate their quality score. The iron torrent run, typically your Q is like 17 to like maybe 25. People tend to think that iron torrent puts themselves at a disadvantage because they kind of underestimate their quality score. So the truth is somewhere in between these two technologies right here. But anyway, you get this data back and you have these quality scores, which will be very useful down the stream in terms of doing analysis. Okay, so that's the data format. Is most of them give you a FASTQ score. I mean a quality score in a FASTQ formatted file for the sequence data. I'm sure there are other formats. So just briefly. You remember solid and the crazy two-base encoding with color, whatever? They generate a color space file where it's just red, green, yellow, blue, whatever. And I mentioned that it's a detriment to them because they push out a new data format that people couldn't understand, couldn't work with, and it took maybe a year or more for people to start working with them. And so there are options where you can map in color space, so you have a reference that's in color space. You have your sequence in color space, and then there's an option where you can convert this to FASTQ, and most people would rather work with that. So there are other formats out there that may or may not be well supported, but FASTQ is by far the industry standard. So that's FASTQ. So now, yep.
Sorry, the higher the score, the better, right?
The higher the score, the better. Yep. I think realistically, there probably is nothing out there that can honestly do better than, say, Q40. Q40 will be solid sequencing. With the two-base encoding, they have much more accuracy. I think they can reach maybe Q40. Illumina, I say, would probably start off Q35, Q37, Q38. And then all of these sequencers, if you were to plot the quality versus the run or the length of the sequence, it'll start off high, but they all dwindle like that. And if you were to do some standard deviation, it'll be something like that. So Illumina, I would say around base 70, 80 is when the drop is. Iron torrent, maybe around 60, 70. Maybe with the new chemistries is a little bit better. And the reason why is as you go longer, there's just more noise, things lag behind, right? You have a cluster-based sequencing where not all 1 million molecules are doing the exact same thing every cycle. So you have lagging behind, and that causes noise and error. So it's harder to make an A call here than it is to make an A call over here. Because this is so much cleaner and dark background. This is not by the time you get all the way there. So the Q scores will vary, and typically if you just did this and just cut the first 50 bases, you have pretty accurate sequencing in most cases. When you start dealing with over here, that's when things get a bit more erroneous. So you get your data back, and all it is sequence name, something like that, then your sequence name, sequence name, sequence. You can't make much sense of this, right? This could be 100 bases, and typically if it's I forget how it's encoded, but maybe R1, R2, forward and reverse read, if you pair them together, you have forward and reverse read, it's 100 base this way, 100 base that way. If you just sat there and stared at it long enough, it still wouldn't make sense, right? It's just A, T's, C's, and G's, unless you have memorized that sequence that it belongs to KRAS exon 2, you wouldn't be able to make sense of it. So the very first thing, well, you need to map it, right, back to a reference genome. So, but before you can do that, you want to do several sort of metrics and quality checks on there. First thing you can do is do a quality check in terms of, how long are my sequencing, the quality score, the GC distribution of my sequencing to make sure there's no bias. All these sort of metrics that you can basically run this file through very quickly, and you get an overall sense of how good your sequencing was. A run on on the high-seq today might be completely different on a run the same machine tomorrow, right? Depending on who's operating it, what reagent lot you use. Same thing with ion torrent. The run, someone who loaded it today versus someone who loaded the next day, and whether it's a 100 base pair kit versus a 200 base pair kit, you can get different quality. So you want to check first to see whether it was a good run or a crappy run. And that's why you want to do an overall quality check of your sequence. And so you can look at, if I'm sequencing 100 paired in, do I always get 100 paired in both directions? My Q-scores, do they peter out at the 10th base or do they go out very long? You can look at that. Am I always sequencing a certain thing, GC content-wise? And that's what that's for. And there's other metrics as well that I won't get into. So let's say you pass the quality check. Next thing you want to do is do a duplicate analysis. This should be pretty standard, but it really depends on your application. If you're doing a PCR-based assay, it doesn't make sense to do a duplicate test because everything you do will be duplicated. Everything starts on the same start site and ends on the same stop site. So a duplicate check is to do this. So you want to check, let's say this is where you're targeting something, right? You want to make sure that your sequencing is starting kind of randomly and ending randomly, like that. Whereas a bad run, let's say we do whole exome capture or the 500 gene capture. Let's say you have things like this. They're always starting on the same site. This is an important concept because this is what you don't want. This is what you do want. In a PCR-based assay, you're going to get this no matter what, right? Because I'm only targeting one amplicon versus my target. But in a shearing-based method where you're doing whole genome library construction and then you're doing capturing, you want some representation like this. Because this means that you sheared randomly, your library ligation was random, and then the PCR enrichment steps that you perform prior to capture at the end of library construction, and the one that you perform after capture to do enrichment was not biased and was complex. So if you have something like this might mean that you were only targeting a small subset of your alleles. So let's say you start out with 1,000 alleles here. But in the end, you only get 1, 2, 3, 4, 5, 6. It looks like you only targeted 6 unique alleles. And these alleles, during the final PCR step, was amplified because you used PCR. Does that make sense? So when you do capture, let's say you start out with 1,000 and you captured 50 of them. And then you do washing, you lost most of them and you were down to say 10 or something like that. And then you lose some more, you're down to 6, and then you do your PCR amplification and it brings it back down to 1,000. And then a subset of this, during sequencing, 500 of them get sequenced, okay? So what happens is that if you have allele frequency down to 1%, or even let's just make it easier, 5%, you would expect that your mutation would occur about 50, right? Is that right? No. 20. 20 times, no, I'm right. 50, right? So you have 50 alleles that will contain your mutation. If you sampled correctly, then those 50 that you captured will probably only pick up what? Maybe 2 reads that show the mutation. And so if you keep losing things along the way, what can happen is that those 2 out of 50 will get lost here, right? And then now all you have is wild type only. So what you want to do your best is maintain complexity throughout the entire process. So if this is the case, you would probably want to capture as much as possible. Capture 500 of the alleles, of which 25 will show your mutation. Minimize the loss during washing and maybe maintain 200 along the way. And in the end, you might get back 10 out of 200. And then this is what you can amplify up, and let's say you amplify it up back to 1,000 during PCR. Now you get back up to 50. But this now, when you sequence, will look more like this right here. Because you have less duplicates. You didn't lose it along the way. And you don't have artificial duplication due to PCR. So this is an important step to do. And typically for a next-gen library, when you're doing like exome capture, you probably want this to be less than 15%. Maybe no more than 20% or so. Because this will attest to a good quality, good sampling assay that you did in hopes of capturing down to low allelic frequency and being able to capture the complexity of your library that is represented by your cancer or your disease cells. For germline, it doesn't matter so much, right? Why? You have 50%, right? 50%. It goes back to first lecture about sampling and germline mutations versus cancer. But still, it'd be nice to maintain a complexity anyways for germline. And then duplication rate is also is a variable, is a factor that depends on other things. It depends on how you shear. Because if you only shear like in bulk kind of ways, it doesn't allow you enough chance to create random start sites. It's a factor of shearing. It's a factor of coverage. So if you sequence high enough, right, the duplication rate will increase. You will be able to show that there's high duplication rate here. So if you sequence to 34x coverage, you can probably get a very complex library. That same library, if you sequence to 1,000x fold coverage, then you start getting duplicates of each one of these as well. So it just depends on the sequencing level as well. And I don't have any magic formula for this, but that's the way sort of it is. And then the one final thing is also the target size. So if you're targeting a large exon versus a tiny one, you're going to get way more duplicates here versus here, right? More chance for random things, less chance for random things here. So duplication rate is good to analyze. Let's say you do it and, yep, you get good duplication, low duplication rate, good complex library. Next thing you want to do is called trimming. So trimming can be done a number of ways. You have your read here, right? And let's say I have very poor quality. Let's draw that graph again. Good quality, bad quality. And this is 75th base. This is the 100th one. So you can trim a number of ways. Let's say I want to say that if there's anything bad about the ends, usually the 5' end is going to be pretty good, right? That's where you start sequencing. But the 3' end is going to be bad. I want to do a Q, I think 13 filtering. I think Q13 is roughly a 5% error rate. Any bases here that have a 5% error rate and above, I'm going to trash. So I'm going to draw the error rate, okay? Let's say that's 5% and this is 10%. As I go further along the line, I have very erroneous bases here. So what I'm going to tell the computer to do is take all my sequences, find out where it starts becoming 5% or more erroneous, and basically just trash the ends. So trim it, cut it away from me. So now I'll end up with just 75 bases that I'll deal with. I'm still happy with the 75 bases. Because it's not worth mapping this. It's going to map erroneously, for one. And two, when you do allelic mutation calling, it's going to give you a bad mutation call anyways. Chances are 1 out of 20 that's going to give you a wrong call. So you might as well just cut your losses and just deal with the good part. So quality trimming is one way. Sometimes when you sequence, let's say this fragment is a little bit smaller than what you anticipated. You started sequencing on this side. Whoops, you read into the adapter. So you're going to have a little bit piece of adapter there. So you can do adaptive trimming as well. Cut this away. It's an adapter. It's not part of what I'm trying to test or analyze. So just trash it for me. And let's see. I'm going to do it by size or length. Let's say you do some sequencing and you get, after trimming and everything, right? After you trim for adapters, after you trim for quality, you end up with something that's like 15 bases in length. You've cut out so many things. Maybe I just want to get rid of everything that's less than 20 bases. So you can do it that way. Because when it's less than 20, it's going to start mapping everywhere in the genome. It's not unique enough and complex enough. So you want to do a size trimming as well. So these are some of the standard ways of doing trimming. And I think up front of this, I forgot to mention that you need to do demultiplexing. So demultiplexing should go ahead of trimming. You do your QC checks, right? You do your duplication check. You want to demultiplex before you trim. Demultiplexing is basically doing the barcode sorting. So when you run desequencing, in order to get more out of your run, let's say you want to do 50 patients in one run, the way to do it is during library construction, you're going to use adapters that have a certain barcode. So let's say the barcode is built into here, barcode is built into here. So for patient 1, you want to use a barcode number 1, and barcode number 1 could be ATCGCCA, something like that. Barcode number 2 could be ATGCCGA. Oh, did I do the exact same thing?
Yeah.
Almost. T. There, that's different enough. So it tells you that these need to be unique and different enough you can distinguish the two. So you would do their library construction capture separately, and in the end qPCR and then mix the two together into one tube, and this is what you're going to take to do sequencing. So when you get the data back in that FASTQ file, everything is mumbo jumbo, right? You have some proportion of this, some of that. Hopefully it's half and half. And then you tell the computer to say, take the first 1, 2, 3, 4, 5, 6. That's a 7. That's a weird one. A 7-base pair barcode. Read the first 7 bases, and if it belongs to this, please sort it into this group here. If it's that, please sort it into a second group here. And then now, from one file, you've generated 2 files, and then you can do the trimming that way. You don't want to trim before you demultiplex because who knows, something might happen to the 5' end, and you might trim away your barcode, and now it's not recognizable anymore. So you want to do this first. Okay, so then trim. Now you're ready to do mapping. So mapping is mostly for what we call, I can erase all this, right? There are 2 modes of analysis that are typical. There is de novo, and there is resequencing. De novo was what we did in the Human Genome Project. We had no reference. We had no idea what the human genome looked like. Let's take your favorite critter today, right? From the moon, someone collected a critter. You have no idea what it is. It looks like something on Earth, but we don't know the sequence. We sequence it de novo, we piece it together, and we find out, after piecing it together into contigs, oh, it looks like a roach. So it's a roach on the moon. So that's de novo sequencing. But you don't know what it was before you started with, right? Resequencing is, I know it's a roach. I'm going to sequence it, and I'm going to map it back to what I have as a roach reference genome. Does that make sense?
Yeah.
Okay. Bad example. But you know what you're doing. I'm sequencing a patient. You know, the patient is human, hopefully. And I know that the patient should look some way relative to the normal reference sequence. That's resequencing. So for all clinical sequencing, it's going to be resequencing. For all novel discoveries, brand new organism you don't know about, that's de novo sequencing. So we're not into this business here. That's more for research and discovery. We're into this business here. So what you need for resequencing is a reference number. So for the human genome, there's HG17. I think there was an HG16 as well. HG19, 18. We're now to HG19. It's the current version. These part you don't want to use anymore. This one, still useful because it's very well annotated with all of the information that you're looking for. HG19 is probably the better one because it's going to be better supported. Probably includes data from the 1,000 Genomes Project, et cetera, et cetera. So this is the one you want to use. So whenever you report out a coordinate on the genomic level, it's absolutely necessary to report out the version or the build that you're using. Because if you take a coordinate here, it has no meaning in here. So there are some conversion tools that you want to do, but you want to take this one. You can download from NCBI, from I think the Bro GATK has a version as well. Ensemble has one too, but you want to use HG19 as your reference. So when you say, I map my data to HG19, people are going to know what you're talking about. You take the reference, and you're going to put it into your system, right? Your cluster that you built. So now, the tools. There are hundreds of tools out there, maybe 20 different mappers. There's commercial solutions, there's freeware, there's probably some that are good. I'll just throw out some names. BWA is a very popular one. The Broad uses it extensively. This is sort of the industry standard. What it stands for is the Burroughs Wheel Aligner. And I will leave it up to you if you really want to figure out what Burroughs Wheel transformation is to go onto this website and you'll find out what the algorithm is. And you can pull up the paper as well, because I'm in no capacity to explain the algorithm. But this is the most popular one. It's fast, it's pretty good. It's good for indels up to a certain point, and it doesn't take too much CPU memory to run. 3 gigabytes, I think, is all you need for an HG19 reference. There's Bowtie. There is BFast, there is Mac, there's TMap, tons. CLC Bio, Partech, DNA Star has part of their own proprietary. These are the commercial solutions, by the way. Soft Genetics NextGene, Mini, right? So this is the most popular open source one, and I've used CLC. I have it because it's free, and we got it as part of the prize, so I use it extensively. NextGene I like as well. These two I haven't really liked too much, so that's just to give you a flavor. There's many tools out there. So let's say you pick the one that you pick. So the question is, you have a reference genome, and you have this piece of DNA, and you basically are asking, where does it belong in my genome? So most people, the way they do it, at least CLC, the way they do it, is you basically take this and you set certain criteria to say this matches my thing or not. And it really depends on what you're trying to look for, right? So let's say you're only trying to look for some SNP right here. So it's an A to a T, for example. So not a huge change here. And let's just make it easy. This is a 100 base read length. So you can make it very stringent. I need this to match 100% to my reference. And it'll do that, right? It takes a computing power to put this into RAM, so you have the entire reference in RAM, and it's going to do some sort of way to say whether or not this matches somewhere along the 3 billion bases that's part of the genome. That's a very difficult question and problem, but the computer will do it for you. But this is way too stringent. You don't allow any room for error, right? And you don't allow any room for variation. So if you have a SNP, it really kills this down to 99%, right? You have one base that's different out of 100. So that's way too stringent. So CLC Bio, the way it does, and I think it could be typical with others, is they set a criteria of 80%. And what they do is they have certain penalties. So if you have a base change that's different from the reference, then it's going to take away maybe 2 points. If you have something at the edges that's different, I think if I remember correctly, they take away 3 points. If you have an indel, so not only is a base change, but you have a deletion in here relative to the reference, maybe it's another penalty of 3. So they do different combinations of this, and in the end, they allow for some error, room for some error, but then hopefully 80% of what's left. So let's say you get rid of some of these bases here, some of there, some of here. In the end, hopefully 80% that's left matches to your genome, and that's a pretty good enough similarity to say, yep, that belongs somewhere in the genome on chromosome 2, in this portion of this gene, et cetera, et cetera. And they do that for each read. To make it faster, typically they don't just take this and compare it base for base. They usually make some iteration of this to represent this in a different kind of way. So that's called sort of indexing. And that's what the Burroughs Wheel Aligner does, is it makes this into, so instead of like, I'm making an analogy here, okay? Maybe it says, take this type of sequence, former here, and make it shaped like a diamond for me. Take this type of sequence here, and make it shaped like a circle for me. So it's sort of taking a bigger problem and shrinking it down into a little bit more organized manner. And then take my read and do the same. So this could be a triangle, this could be a circle. And then now do the similarity. Oh, this matches, and then open it up and see what's in detail underneath. So that's how you can get much faster sort of aligning. So that transformation that I talked about, this is a very crude, simplified version of that, but essentially that's what it's doing. You index the reference, you index your reads, and then you do the mapping, and that's how you are able to perform it in a much quicker manner and with lower amounts of RAM and memory. So you do your mapping, and actually the quality scores get figured into here as well. You can say, if you map overall and the average quality is something, please accept it. If not, then throw it away. So there are ways of doing that as well. So let's say you do map. And then now, this is your reference. These are your exons. And here are your reads. Determine that each one of these meet a minimum criteria of 80% match or whatever. And then the penalties are not so high that, yep, I am able to map it. But these are my different reads are mapping. So this is called a pileup, or you can use something called the Integrated Genome Browser. A viewer, to see this. You can use UCSC Genome Browser can also take, I think, a BAM file. If you can upload it and actually view it on there as well. Or you have some other proprietary thing, like CLC has its own browser or viewer. So you can view it this way. But right now, if you do this, you can also want to layer on top of this some other additional information. So what you can do is just draw lines straight through here, right? And basically count up how many reads cover that one base. So this will give you a coverage plot. Like that. So that's coverage. So coverage is, I take this 1A, for example. How many reads cover that 1A? I have here 100 reads that cover that 1A. So that's 100x full coverage. In the peak here, I have something that is 200x full coverage. So it's variable because during selection, you don't have the luxury of being able to pull down something specifically. You're left up to the will of the assay and the biology that's associated with the hybridization step. So this is what you're going to get. So sometimes you get great coverage, sometimes you get poor coverage. It's just a design of the assay that you have to take and tweak it to make sure it's correct. So perhaps you see this on the first one, you say, okay, let's boost up the baits for this in SureSelect. And let's boost it up so that I can get a little bit better coverage. So that's sort of a trial and error kind of thing that you have to do. And so you have sufficient coverage now. And the question is, you know where it's mapping genomic-wise, but you really have no idea what this is, right? So the next step is to do annotation. I guess you could do this last, actually. So you can do annotation up front if you wanted to. Let's just say that we annotate it. So what we annotate is that this is the KRAS gene, and this is exon 1, exon 2, exon 3, exon 4. There are free tools that are available that can give you the annotation. And this could be many, many different levels of information. So let's just say we annotated with the gene name, the exon number, the promoter region, the UTR, non-coding RNAs, microRNAs. You can annotate to many, many different levels. It's just basically saying, what am I looking at here? So here I'm looking at exon 2 of KRAS or exon 1. So that's annotation. When you're ready, after you mapped, then you want to do variant calling. Because now that you've mapped, you're going to scour through your entire read mapping. You can say, what is different between the reference and my patient sample, my read mapping? And that's called variant calling. You can do SNP calling. You can do indel calling, and you can do copy number. There are tools that you can do that. Some tools can do rearrangements. Let's just stick with these two for now, okay? So SNP calling and indel. Indel is a bit tougher to do. So you remember I say 80%, right, has to match on. So here doesn't match. 80% has to match. If you have a deletion that's quite large, that already kills quite a lot of your capacity. So there are certain other aligners that are better at this. So certain other aligners really start mapping on one end of the read. So, sorry. This is your read. They maybe map this in first, they map this in separately, and then they move in from there. So it's a little bit better algorithm. A bit more complex, more computation intensive, but these are designed to better map indels. But other ones like BWA, sometimes they don't perform so well with indels. So that's a little bit tougher. SNPs are easy, right? So let's say I have a SNP here. I have several reads here that are showing A, but my reference is a T right there. So you do your variant calling, and it can spit out the frequency, how many reads show an A, how many reads show a T, what is the reference. And thereafter, when you annotate it, you'll know now that, oh, it's KRAS, it's in exon 1 at a certain position, it's a T to an A mutation. Thereafter, you want to annotate even more. So you want to annotate, is it synonymous or non-synonymous? Is it coding or non-coding? I mean, I guess it's intuitive here, right? That it is coding. You do a coding region or non-coding. Synonymous or non-synonymous. Is it a splice junction mutation? Let's say it's in a splice site. You can do these types of annotation. What is the amino acid change that you're getting? That's another level that you can do. And let's say, have I seen it before in Cosmic? You can annotate that way. Do I see it in DB SNP? Is this a SNP that occurs very frequently in population? For example, EGFR has a very common SNP that we don't worry about whatsoever. But if it's not a SNP, and it is in Cosmic, and it has been shown to be causative of disease, now via an annotation, complete annotation, you can go through and make a diagnosis. It's very different, right? Very different from traditional sequencing. Traditional sequencing is, you have your blips. Sorry, it's monochromatic today, but here's your mutation. And you say, oh yeah, there's something different from reference. I see it both in the forward direction, and it's crazy, but in the opposite direction, they're the exact same color. You see in the reverse direction, and then I call my mutation. This, you can't, I mean, you can visualize it if you want, but if you deal with 5 million bases, you let the computer do that part, right? So in the end, what you want to do is put all of this together into one file, and that's going to be called the variant call file, or the VCF file. It's a tabulated, tab-delimited file, tabulated with the genomic coordinates, with all the annotations that you need, the number of reads that cover the reference, the number of reads that cover the mutation, the quality scores. You can build into here a quality score as well. Like, let's say during variant calling, I don't want to call this unless the average quality score is Q20 or Q30. You can set it whatever you want. And that way you can get rid of all of the erroneous reads, etc. So if you do that, you end up with a VCF file. So no more is this, where you have to look at the analog raw data. It's going to be tabulated, basically almost digital, the data that comes out. But the practice is the same, right? In the old days, we have a reference. We have forward and reverse read. We have to align it, right? You have to align it to what you think you're trying to read. And then you make the variant calling here. And then you have to say, oh, what base is it? What mutation is it? It's all the same process, but you have to now let go of that and let the computer handle all of that, which is a nice thing, right? And so that's the main difference between interpreting a Sanger sequencing trace versus next-gen. But really, in the end, it's the same process. So now that you get the variant call file out, well annotated, you need to filter. Because you're going to get things where I have very low coverage, and my sequencing is very erroneous. So let's say half of these reads here show a C, and half show a G. Now that you get the variant call file out, well annotated, you need to filter. Because you're going to get things where I have very low coverage, and my sequencing is very erroneous. So let's say half of these reads here show a C, and half show a G.